Well, good morning, folks. Here we are. It's time for yet another class. So I think we're kind of just hopping right into the good old midterm season. And I imagine you're all fairly busy with that type of stuff. That's one of the great pleasures you have as students is you don't get to just learn, but you also get to be tested. Unfortunately, that's an administrative reality that we haven't yet overcome. For some reason, we are obliged to put a number next to your name that says, how good you are or how worthy you are of success. And, we haven't yet come up with a solution to that. I'd love to just skip the whole thing and just talk endlessly, but hey, this is how she works. So today's lecture is going to be partly some new content. I'm going to just tell you a little bit about how scientists working in the genomics field are trying to link genes and variants of genes to traits using genome-wide association studies. And then I'm going to go through and just do a bit of a review for about the second half. So stay tuned. If you have any questions and you want to think about them, please kind of get those ready. Maybe we can go into those in the second half. If you aren't here to ask a question, there's always office hours tomorrow. I'll be live broadcasting from Zoom, and you can ask a question that way as well. Whatever works for you folks. So without any further delay, let us begin. So today we're talking about linking genes to traits. The overall goal of this class is to not just show you this today, but to give you kind of a more grounded understanding of this, because essentially the whole class is about linking traits to genes, the traits being mental illnesses and the genes being whatever they're linked to. So this is not going to be all inclusive. We'll be talking about various elements of these topics in a more practical way as we go in and talk, and discuss different disorders and so forth. So this is just kind of an introduction that I wanted to slip in here just to make sure that you're all on the same page when it comes to understanding genome-wide studies. So as I've already kind of discussed, there are a variety of ways you can approach the question of how do you link genetics to phenotypes. Phenotype being anything from normal variations in traits, such as height or weight, or perhaps even personality, kind of continuous variables that can be at a number of different stages, or hair color or whatever, all the way up to and including disease states, mental illnesses, neurological conditions, and so forth. And there's kind of two broad ways to go about this. You can either start from a particular hypothesis where you think that some gene or some pathway or some variant might be related, and grab a bunch of people and study it study just that particular area. These are kind of the a priori candidate gene methods. And I think we've established that those are fairly weak, and for that reason they're not done very much anymore. But the other alternative would be to look across the entire genome and see if you could identify on a genome-wide level what is there that's correlating with the trait that you're interested in. And of course, the strength there is you don't have to know ahead of time what you're looking for. You just it's like you're sort of like shaking the genome and seeing what falls out. And whatever treasures you get from that type of study, those are going to be a bit of a surprise to you. However, that's where you really find the new interesting biology. It's a bit like how some people might, you can certainly have a lot of fun pointing a telescope up into the sky and looking at Saturn. That's great. But you're not really discovering anything. You're just observing something that everyone already knew was there. It's not easy. You have to know where Saturn's going to be and so forth. But, broadly speaking, you're not going to discover anything that way. The real discoveries are made by scanning the sky in a kind of random or unbiased way and seeing if you find anything new for every declination and every ascension and so forth. Anyway, so this is the aim of the GWAS studies, and prior to the GWAS technology that we're going to talk about, the method was a genome-wide linkage study, which is kind of, looking at it now from the hindsight of the genomics era, it seems kind of absurd how these studies used to work. We'll talk about them a little bit in the schizophrenia lecture, I believe, but for now, this is just, suffice to say, this is a very old and imprecise method that would allow you to link traits to kind of specific parts of a chromosome. You could kind of link, say, schizophrenia to a certain band, for example, on one chromosome, on the short or long arm of one chromosome, and that's better than nothing. But you're not going to be able to know a whole lot just from that. There's potentially hundreds, if not thousands of genes located within all of these loci. So they were rather imprecise, and more importantly, they were incredibly time consuming. So the amount of things that were discovered there were pretty limited. The current gold standard is called a genome-wide association study, or a GWAS. I know all of you acronym enthusiasts out there are probably thinking, why are we saying GWAS study? That's kind of redundant, right? Because yes, in GWAS stands for study. Well, it's a bit like when you say ATM machine, right? I mean, this is just sort of the way things work in English. Unfortunately, it doesn't make a lot of sense, but it's impossible to, or it's so much more work to not say it this way that I don't think there's any point in bothering. It's kind of like people who use the word data as a singular. It's not correct. Data is plural, but gosh, I mean, that's not a thing to worry about. Anyway. The aim of a GWAS study is to look at the entire genome and try to link variants that you can genotype to traits or to disease states in an unbiased manner. So you're not picking things ahead of time. You're just kind of poking the genome with a stick and seeing what traits fall out. And there's also a version of this that's used in epigenetics called an epigenome-wide association study, or an EWAS. And this one is typically what's happening here is the researchers are taking peripheral tissue, perhaps typically peripheral blood. Blood has lots of DNA in it, so you can measure DNA methylation there quite easily, and looking to see if a given trait correlates with DNA methylation at CpG nucleotide or CpG dinucleotides somewhere in the epigenome. Typically, these are I think these are very weak studies. I don't think they're very good at all. So we won't really be talking a whole lot about them except to criticize them. Although there are tons of them that get done. There's so many of EWAS studies are being done every year. It's like it's quite astonishing given that they're not too informative. But anyway, we'll look into that as needed. So the main thing we're going to talk about are GWAS studies. But I think the framework for GWAS applies quite widely. You're taking the entire genome and you're trying to see if there's associations between anything there and the condition or the trait or the disease you're interested in. So this is kind of a window into the dark pre-genomics era. And really, when it comes to genetics, the pre-genomics era was almost all of the history. The genome era we could broadly define as the post-2000, maybe 2001 era. That's when the human genome draft was first released. This is when it finally became possible to understand human genetic variation across the entire genome in a way that was, well, at the time, in a way that was pretty limited to people of white European descent, but has since expanded somewhat. And so these early studies that came before then, it's like you're sort of picking areas of the genome that you know about that have been studied previously, and you're saying, okay, well, within these small areas, is there any association between a given, between variation in these areas and a given trait? And this is just a little meta-analysis from previous candidate gene studies where researchers would choose a given locus and look there for associations between the variants and the disease or whatever of interest. And so here's a number of them. We're talking, like for each of these different loci or genes, you can look at the number of studies that were done. This is again pre-genomics era, pre-2003. This is when the review was published, or the analysis was published. Like 4 studies done here, 12, 20, 48 studies on the relationship between DRD3 and schizophrenia, and so on down the line. And what they're doing essentially in this meta-analysis is looking to see, okay, of all the studies that have been done that meet some minimum quality threshold, if you put them all together, add them up, do they show any kind of effect at all? In other words, does the association tend to replicate across all these studies when you factor in, when you kind of control for how rigorously they're done and how much potential error exists in each study? And the answer is that for the vast majority, or not, well, I guess for the majority, maybe perhaps not the vast majority, but for the majority of these variants, they don't actually end up replicating when you look at them in a bigger picture. So, there are, for example, 7 studies linking variations in the gene NTF3 to schizophrenia. When you put them all together and kind of look at them in as unbiased a way as you can, they make absolutely no difference. So the odds of having schizophrenia relative to whether or not you have this sort of the risk allele are 1.01, but the confidence intervals overlap 1. In other words, statistically speaking, this variance in this locus make absolutely no difference. And so you can say like some of these do work. Some of these did end up working. DRD3 is a dopamine receptor subunit. So that's I don't really have any. I don't think there'd be too much controversy to say that that would be involved in schizophrenia, given that it's related to dopamine, as well as the HTR2A. That's a serotonin receptor gene. That's all well and good, and there's 28 and 48 studies respectively. But so many of these don't replicate, and just were essentially a waste of time. Maybe that's a bit strong, but they just turned out to not work. So all that research, even if the researchers had reported positive results when they published it, when you put everything together and look at it from a distance, it doesn't really pan out. So this is what you would get with candidate gene studies. They had small sample sizes. They tended to be rather biased. They tended to not replicate very well, meaning that if you looked at the same region in a different population, or if you did a study in your own hospital, for example, they just didn't come out the same way. So things seemed pretty hopeless. But now we're in the post-genomics days, the brighter days, where after the completion of the Human Genome Project, as well as projects like the International HapMap Project, which characterizes basically linkage disequilibrium genome-wide, researchers are now able to actually kind of grasp the full scope of human genetic variation, and thus the extent to which this full scope correlates with traits or diseases that they're interested in. The tool of choice is what's called a SNP array. These are microarrays, and we talked about microarrays briefly in the sequencing lecture, that are able to genotype individuals for millions of SNPs at one time. So these are fairly simple to run. They're just basically a chip, about the size of a microscope slide, and roughly speaking, you just kind of throw the person's DNA on there. Wherever they have a SNP, it'll stick to the microarray, and you can detect whether what their genotype is for all of those SNPs. And you can do this for millions of SNPs at a time, and thus collect very detailed genotyping information from people without a whole lot of hands-on lab time. This is still much quicker than doing sequencing for everybody. And this does give you a fairly accurate picture genome-wide. And what this can let researchers do is they can go beyond these a priori hypotheses where they have some particular relationship in mind and screen the genome for unexpected relationships. This is what a genome-wide association study is. So GWAS studies, and again, this genome-wide association studies. That's just what it translates to. They're designed to relate genotype to either a trait or a disease state. So I think this is kind of worth dwelling on a tiny bit. There's basically two kind of categories of designs, or two kind of ways that you can design these studies. If you're talking about a disease that's sort of a binary condition where you either are a case or a control, meaning you're either somebody who doesn't have the disease, you're a control, or somebody who does have the disease, you're a case, you're making kind of a dichotomous comparison, comparing one group to the other. Other conditions, or other traits, I should say, are rather more continuous. They would be things like height or BMI or waist-hip ratio. I'm just thinking of all the anthropometric ones, but they're any trait that can be described in a continuous way. These are two different ways of organizing the study, and I think maybe just as a small side point, there is something to watch out for when you're reading research, and this goes beyond genomics, but just would apply to any research, is you should be very careful to look for when researchers sort of arbitrarily dichotomize a trait, because that potentially would mean that they're trying to pull something funny. So, if you have a study, for example, where you're trying to see if the dose of a given drug correlates with obesity risk. And the way you would probably design that is do like a dose response curve where you have the dose of whatever you're giving them on one axis of the figure of the scatter plot, and then the body weight on the other, or BMI or whatever you're measuring on the other axis of the scatter plot. And you would try to draw a regression line through there. And if the regression line had a fairly steep slope and it had a good R squared, then you would say there was a relationship between the two things. And researchers would, I think, tend to want to use that model, but oftentimes what you'll see if you're reading a study where that would be the logical way to analyze the data, sometimes what they'll do is they'll just dichotomize it and say, okay, well, that didn't really work. Let's just split them up into obese and non-obese. And they just make it a cutoff line somewhere, and they just lump them into two groups, and then they can run the analysis as though it's a case control study. And of course, whenever you dichotomize a continuous variable and break it up into chunks, where you make that breakpoint is very subjective. And if you move it around, sometimes you can get better or worse results that are, I guess, more or less consistent with your hypothesis. That's not something that's just something to watch out for when you're reading research in general. This doesn't exactly apply to genomics, but I thought I would bring this up because it's a common, trick that's done by researchers that can make things look, basically a way of reshaping the analysis to provide them the results they're actually looking for. So stay tuned for that. It's a very, very tricky thing to do. It happens all the time. Many such cases, folks. Anyway, so with a case control study, what researchers are typically looking for are to determine an odds ratio for each genotype, meaning that for every copy of a given allele that you have, your odds of having the disease are increased by a certain amount. Whereas trait-based models, because you're working with an unselected sample and just get a continuous population, you're really tending to try to build regression models. These are going to be less common for our world because we're mostly talking about mental illness, and those use case control designs. But a regression model would just be sort of trying to say, okay, so for every copy of this allele you have, how many units of BMI do you gain? So, if you were to find a positive correlation there, or, if you had a positive regression coefficient, what it would mean is that allele seems to correlate with obesity in a way that might be causative. So this is all done in standard deviation units, so in Z-transformed units, but this is sort of the other way that things can be done. And of course, once you have your design, you can genotype people either using SNP arrays or with whole genome sequencing, WGS. And this is this would be kind of the way of the future, but it's still not terribly common for GWAS studies because it's still quite expensive. And if you want to sequence a person's entire genome, you're talking about $1,000 a person. Roughly speaking. So if you're doing a study where you need thousands and tens of thousands of people to achieve statistical power, you're not going to be able to afford that, at least not yet. So I wanted to give you just kind of an example of the mathematical logic used in case control studies. And this isn't to say that this is a math class or that I expect you to be able to work this out on paper. It's more just like, for some people and the way that some people think, not me, by the way, but some people, seeing it kind of written out makes the concept a little more real. So I'm going to try to present this to you and maybe it might help you understand things. So this is the method known as allele counting. This is only one way that variance variance can be linked to a case control study. There's other sort of methods you can use to do this. This might be the simplest, but I think it gives a pretty good sense of what we're doing. So imagine you're a researcher who's running a GWAS study and you're looking at schizophrenia and you've got a sample size of 10,000. 10,000 would be a pretty normal-ish sample size for a question like this. The more, the better, of course, but 10,000 isn't too bad. And typically in a study like this, 1% or so of the population might have schizophrenia. So typically you're going to have fewer cases than you will controls. Maybe we have 2,000 cases and 8,000 controls. And then you go through and you're genotyping all of the SNPs. And the genotypes, of course, for every variant would either be, like let's say this is, I've just made up this SNP, it's a fake SNP, but let's say it's located in an intron somewhere in this imaginary gene. And the genotype... The genotypes could either be AA, AT, or TT, meaning that they might have, they might be homozygous for the A allele, or heterozygous, or homozygous for the T allele. And then you have this information. This is what the SNP array is going to give you. And then you count up, for the cases and controls, you count up the number of people who have each allele. So let's just say these are the numbers. You see that the cases have 1,204 people. 1,204 people who are cases have the A allele, whereas 2,789 have the AA allele in the controls. And you kind of go down the line and you just count up the number of times everyone in the case and control groups has any of these genotypes. And then you sum them up. So you count up the total number of A's. So you would take, in this case, 1,204 plus times 2, because there's 2 A's here, plus 523, That gives you a total of 2,931. And you would do the same for T's and also repeat that in the control group. So this gives you the total number of A's, copies of the A allele that are in the case group, and the total number of copies of the A allele that are in the control group. And that's really all you need. Now, just look at this kind of visually, and what you should be kind of seeing is that there's way more of the AA allele in the case group than there is in the control, relatively speaking. There's way fewer cases, there's only 2,000, but close to, or over half of the cases have the AA allele. Whereas when you look at the controls, there's 8,000, and maybe only way less than half have the AA allele in the controls. So kind of right off the bat, you can kind of see, okay, so the AA really seems like it's overrepresented among the cases, the people with schizophrenia. That would give you a sense that maybe it has something to do with schizophrenia, because the people who have schizophrenia have way more copies of this AA allele. So the odds ratio for this particular thing, given that A would be the risk allele, is just saying, okay, what are your odds of schizophrenia if you have the A allele, divided by what are your odds of schizophrenia if you have the T allele. So to calculate this, you would just calculate the odds ratio for A, where you take the total number of A's in your case, and you divide it by the total number of A's in your control. So 2931 divided by 6698, and then you would take that and divide that by the total number of T's in your case, divided by your total number of T's in control. So this is what it would look like. You're just plugging these numbers in, and then you kind of do your, you push the equal sign on your calculator, and this is what you would get. So the odds ratio for A is 3.81, meaning that, very roughly speaking, for every copy of the A allele you have, your risk of schizophrenia is 3.81 times higher than control. This would be a very strong effect. This is, I made these data up, they're fake, but this would be a very strong effect. This would mean that if you were to genotype somebody and look at this allele, you could guess reasonably, you'd have a better chance of guessing whether or not they're going to be diagnosed with schizophrenia. This is quite a powerful allele, and whatever gene this is located in, you'd really want to know what that gene was and where it was expressed in, and everything else along those lines. So that's just one example of what we're talking about here, but essentially with a case control study, you're trying to compute the odds of having a given condition relative to how many copies of a certain allele you happen to have. And of course, this math would be repeated for every single allele in the SNP array. So I just got a question here. So could you repeat how the 3 genotypes in the table were determined? Sure. So these 3 genotypes are coming from the sample population. So what's happening is you're using a microarray, which is a tool that can measure somebody's genotype at a bunch of different locations, and you're just genotyping people. So the 3, for every given SNP, there's typically, 3 states you could be in. There's 3 genotypes for every SNP, typically. There's homozygous for the major allele, which is the more common of the 2 alleles. This might be TT, for example. There's heterozygous, where you would have 1 copy of the major allele and 1 copy of the minor allele. And then there's homozygous for the minor allele, which is the less common of the 2 alleles in the overall population. So the genotypes are just given to you by the molecular biology, by the tools that we actually use to make these measurements. And then this comes to you as basically a spreadsheet. So for every row of the spreadsheet would be a SNP, and then every column would be counts of a given genotype in your cases and controls. Does that make sense? Okay, so while I wait to hear if that made any sense, I'm going to move on to the next slide here. So if you've taken any statistics before, okay, so the microarray determined it. Yes, Natalie, it's determined by the microarray. This is just a tool that genotypes people. It can genotype people for millions of different variants all at the same time. So it's a powerful tool for genome-wide genotyping. And there's a number of steps involved in doing it, but ultimately the result is just for every individual that you have in your study, you get a genotype for every SNP. You either get AA, AT, or TT in this case. So that's the type of data you get. And so the aim in processing the data is to simply go through and for every SNP, try to figure out if a given allele is more common in the controls or the case, or well, more common in the cases than you would expect given the kind of distribution in the population. Anyway, so ultimately, if you remember your statistics classes or if you've taken any, you may recall this problem of multiple testing. The statistical significance of any given test is typically given as a p-value, which is roughly described as the probability that a given observation could be made by random chance. So, we can all, if you look at enough stuff, something is going to seem significant, something's going to seem different. So you kind of have to have a sense of like how likely would it be that you would make that observation if the data were truly just random. And that's given by the p-value. I think I don't need to get into that a huge amount, but I would say that if you haven't learned about statistics, I think it's a pretty good idea to take some stats courses. The cutoff for p is typically less than 0.05, which means that there's about a 1 in 20 chance of, or a 5% chance that the results you've observed, there's a less than 5% chance that the results you've observed arose by chance, or given that the null hypothesis could be true, there's less than 5% chance that you would observe those results if the null hypothesis were true. So that's kind of the formal definition of it. The way researchers would use this is they would just make a bunch of statistical comparisons, and then any comparison that was significant, they would put a little star over it and say this is a meaningful piece of information. And that's all well and good. The cutoff of less than 0.05 is certainly controversial, but it's the one that everyone uses. However, again, if you recall from your stats classes, this is only if you're doing one single test. If you're just doing one t-test, that's the cutoff you can use. However, if you test lots of things, this starts to become, the significance tends to become inflated, because for every individual test, there's about a 5% chance that you would get those results purely by chance. And that's a level that we can tolerate on, for individual tests. The stakes aren't that high. But if you start making millions of tests and you're talking about 5% of them being significant by pure chance, that's going to lead you in all kinds of horrible directions, because it's inevitable. If you do millions of tests and there's a 1 in 20 chance that you'll get a significant result purely by chance, then you're going to get, thousands and thousands of significant results that would be biologically completely meaningless, but would have the appearance of being significant. And this could lead you in all kinds of bad directions. So the way that this would be approached is with correction from multiple testing. And the logic of this is the more tests that you run, or the more statistical tests that you run, the lower your p-value cutoff has to be. Again, this is something that you can save the details of this for your stats lectures, but essentially you're trying to adjust the p-value cutoff so that it's fair relative to how many tests you run. And SNPs, of course, SNP arrays and GWAS studies are looking at potentially millions of tests at the same time. So you have to deal with this somehow. Let me just stop for a minute and point out what this is. This is what's called a Manhattan plot. The reason it's called this is because the kind of look of it looks a bit like a city skyline. You've got kind of skyscrapery type things. You've got shorter buildings, and it looks, like Manhattan, kind of like a city skyline. Whatever. It's kind of a special variant of a scatter plot, but what you have here are chromosomes along the x-axis. So all this brown one points are chromosome 1, and then 2 and 3 all the way down the line to 23, which would be the X chromosome. You can see the chromosomes get smaller the higher the number. They're just numbered by size, except for the X chromosome. Anyway, so that's what is on the x-axis, and then each point represents a given SNP that was genotyped in the study. So every point is one of the millions of SNPs that the researchers would look at, and then the height of the points are the negative log 10 transformed p-values. So if you negative log 10 transform, basically the smaller the number is, the higher it is after the higher it becomes after you negative log 10 transform it. And the reason they do this is only to make the plot more visually appealing. If you have on a regular scatter plots axis, the difference between 0.001 and 0.0001 that's that's an order of magnitude difference, but it would look like a microscopic amount on a regular axis, because you're talking about such a small range. So the purpose of transforming the axis like this is to kind of spread out the range and give you a better separation between the low p-values and the really, really low p-values. So the really low p-values, the SNPs that are very significant, end up being higher. And so the problem of multiple testing, as I was saying, would typically, or technically, make it impossible to discern signal from noise in a GWAS study design, because you're making millions of comparisons at the same time. Like imagine if you're imagine if you're like a fortune teller and you're saying, I can predict your future, or maybe I can guess your body weight, or I can tell you who you're going to marry, whatever it is that you might claim to be able to do. If you got one guess, it would be very impressive if you were correct about something. But if you got a million guesses, it would be inevitable that you would get some right. And in fact, this is kind of how these types of people would tend to work. Cold reading is a way of acting as though you're able to fortune tell, or able to see into the past, or read people's minds or whatever. You just keep saying stuff and then wait until you say something that happens to be correct or close to being correct. And then you're like, oh, yes, of course. Yes. So you're saying that you had a sister. Oh, interesting. Was she younger, older? Older, yes, of course. I knew it. She was older. And you kind of just fake your way through. But because people tend to disregard all the ones that are wrong and focus on the ones that are right, it makes it seem like you're very smart and you have this magical power. And this is kind of the issue with statistical testing. If you make millions of tests, you don't even have to be right ever, but some of them are going to come out significant just because. Anyway, sorry to kind of beat around the bush with that one, but it's an important point. One that's not, it's fairly well, acknowledged here in this field, but not so much in science more broadly, even though statisticians love thinking about it. What researchers have done is, instead of going with the normal p less than 0.05 as a cutoff, what they use for GWAS studies is called the genome-wide significance cutoff of p less than 5 times 10 to the negative 8. So that's a much, much lower cutoff, and it basically was determined by doing a Bonferroni correction based on the total number of common, minor allele frequency greater than 5, SNPs in the human genome. So, basically they just kind of took a back of the envelope calculation and said, okay, there's about, this many common variants that exist in the human genome that we can look at, and what would be a fair p-value if we're talking about making that many comparisons. So this is typically just a line that the researchers would draw across the Manhattan plot, so that any points that were above this line were considered significant at the genome-wide level, and any points below it are not significant. So that's the point of that, and I don't. It's not like this is going to be make or break when it comes to your understanding of how a GWAS study works, but I think it's an important consideration, because it's a basic part of statistics that is really quite relevant to many areas of science, and especially something to have in mind when you're making these giant genome-wide comparisons. I guess it's probably also a reason why the candidate gene method tended to be so biased, was because if you're using the candidate gene methods, you're only looking at a couple of different places, maybe one SNP or a handful of different loci, and there the adjustment for multiple testing is so much lower. You might have to only make one comparison, therefore your p-value cutoff can be 0.05. That's very lax if you think about it. 1/20 chance of having a false positive. That's those are pretty good odds, So if you do like 20 different experiments, one of them is going to be wrong just because of this false positive rate, or because of this false discovery rate. And if you publish only the one that comes out significant, you're going to significantly bias the literature. Really, people should be publishing everything, but typically people only publish positive results. Anyway, rant over. So typically, a GWAS study is going to produce somewhat ambiguous results. Now, we can if you find coding variants that change the actual structure of the protein and make a protein non-functional, or change the enzymatic process that it uses or whatever, then you can kind of interpret that, right? If you're looking at an enzyme that is involved in dopamine synthesis, for example, and there's a coding variation that changes how efficient the enzyme is, then certainly you can kind of work with that on an interpretive level. But really, most GWAS studies don't give you that kind of result, because most of the interesting variation in humans is not in the protein coding sequences. Protein coding variants are quite rare by comparison, because usually they're very harmful. It's very rare to get a coding variation that makes a protein work better. Almost all the time when there's a variant or a mutation in a protein coding part of the genome, it makes the protein worse, and it's either fatal or it decreases your reproductive success. So those tend to be selected out unless they have some kind of selective advantage. Just evolution at work. So most variants are not located there, and so they're located in introns and promoter regions and intergenic regions and places where there's no nearby genes. So what you have to do is annotate the genome and add in more information to tell you what possibly could be going on in those parts of the genome. So this is kind of an example of what we would mean by that. You have your GWAS data that would tell you that, for example, there's a SNP that's interesting here, but where is this? There's no gene nearby. Is there anything else that's nearby? Well, you can look at chromatin accessibility. This describes how the three-dimensional structure of the chromatin enables access to the genome from transcription factors and enzymes. So the chromatin can either be condensed and everything can be kind of packed away, or it can be rather accessible, and thus the genes in that area are more easy to transcribe. So that's one thing you can measure. That's sort of a relevant epigenetic feature. There's histone modification. You take your pick. There's dozens and dozens of types of histone modifications you can look at, whether it's H3K9 trimethylation or H3K27 acetylation. There's tons of different histone modifications you can add, and the way these plots work is typically the higher these peaks are, the more of a given histone modification there is in a particular part of the genome. So you can add this kind of epigenetic information to it as well. You can measure DNA methylation. We've seen how you can do that with the whole genome bisulfite sequencing and quantify the amount of methylation to CpGs there is in all these areas. That's a relevant piece of epigenetic information. And you can also just look at where transcription factors bind and say, okay, in this area, the transcription factor DLX3 binds, and over here the one LHX2 binds, and this kind of thing. And these are all pieces of information that help fill in the blanks of what these variants that you've determined might be associated with a disease, what they actually do in the genome. Without this, it's like having a map of the city, but no one's telling you where the roads are or where the McDonald's are or whatever. The kind of like layers of information on the map of the genome. And this, by the way, is what you would see when you look at the genome browser. You see that by default the genome browser will tell you where the genes are. But you can also layer on top of that all kinds of other types of information, where there's CpG islands, what the GC content is, what the conservation level is, all this kind of stuff. And the more information you've layered on top of this, the more easily you can interpret SNPs and variants that you discover that might be related to a disease. So this is kind of our goal here, is on the one hand, we're doing GWAS work to figure out where the variants are that are related to disease. And then on the other hand, we're trying to annotate the genome to find out what is happening there other than that variation, what is happening epigenetically. And by the way, all of this stuff is tissue specific. So you're not just looking in any old tissue, you're trying to look in the target tissue. So if you're looking at, for example, a mental illness, you'd want all these data from the brain, and not just the brain, but particularly neurons, or glia, who knows, but a particular cell type from the brain, from a particular location. So it gets rather complicated. So there's kind of this sweet spot that GWAS studies are able to identify or to really study effectively. So here is a figure from Tam et al. In 2019, and it's a really simple plot. They've got allele frequency on the x-axis. So alleles could either be very rare, meaning very few people would have that particular allele, or very common, meaning it's kind of 50-50 or even more common than that. On the x-axis, and then on the y-axis you have effect size. So a low effect size means that whether or not you have that allele makes almost no difference in your risk of a given disease. So the odds ratio might be 1.02 or something, like a very small difference, almost negligible. All the way up to high, where with a high effect size allele, whether or not you have that would determine almost with complete certainty whether or not you would have the disease. So for example, like Rett syndrome, for example, if you have a mutation in your MECP2 gene and you're a female, you'll basically be guaranteed to have Rett syndrome. And there's lots of ones like that. So it's sort of typically like a monogenic condition where the presence or absence of a given allele guarantees that you'll have something. So these are kind of the continuum, and alleles can be all the way from no effect to very high effect. So if you kind of plot these out and think about all the different variants that you can study, you'll have in one corner rare variants that have a low effect size. So these are very difficult to detect because typically SNP arrays aren't trying to detect rare variants, number one. And number two, small effect sizes require gigantic sample sizes to be able to detect. If something's making a 0.5% difference in your risk of something, you need lots and lots of people to be able to make that determination. So these are very difficult to find, although they might be a pretty important part of the genetics of disease. A lot of small variants, a lot of rare variants with a small effect can add up to having a pretty large effect if you put them all together. But on an individual level, they're very tough to detect. And then over on the other corner, you have common variants with very large effects. And typically the types of diseases we study with GWAS aren't like this because those have already been discovered. Common variants with large effects are very easy to find because the methods of classical genetics can find those pretty well. So GWAS studies don't tend to really look in those areas either. Where GWAS focuses are this kind of sweet spot in the middle here, this band down the middle, where you have common variants with small effects. Common variants are easy to measure because even with a relatively small sample size, you'll get enough people with either copy of the variant to be able to make a determination. Even if the effect size is fairly small. There's also intermediate frequency variants with moderate effects. And then highly penetrant mutations mean that they're rare, but if you happen to have one, you have a very high likelihood of having the disease. Penetration just means like, if you have a given allele, how likely will it actually manifest as a phenotype? So highly penetrant ones, if you have it, you're kind of have a very high chance, if not a guarantee, of developing or having the phenotype in question. So the types of GWAS studies we are used to today kind of focus on this band in the middle, and these are the kinds of things they can discuss or discover. However, there is this kind of like undiscovered world of rare variants with small effects that might be playing a pretty big role in disease risk that right now is kind of like the dark matter of the genome. We suspect that it's there, but it's not easy to detect using our current methods. You would really need whole genome sequencing to find these things, and you'd need bigger sample sizes. And whole genome sequencing on a big sample size, that's very expensive and very time consuming. So there's kind of a reason why we don't have this information yet. But that's kind of where we think all the new good stuff is. We've kind of all this kind of middle area. This has kind of been picked clean. Not for every disease, but for a lot of conditions, I think we've probably found about as much as we're going to find using the normal methods. Anyway, so the last thing I wanted to mention was sample size, and this is another consideration when we were talking about GWAS studies. You need, and this is true of any study, frankly, the bigger the sample size, the more statistical power you have. Statistical power just refers to your ability to detect a effect if the null hypothesis is false. So your ability to detect a real effect, given that it's in your sample. So small sample sizes just aren't that effective at doing this, right? I mean, if you have a sample size of 4, there's so much variation within the sample that it would be very tough to separate it from variation between your study groups or between your control and your case group or whatever. So the bigger the sample size, the more variance you can discover. This is also because bigger sample sizes allow you to identify more low frequency variants that have small effect sizes. These are kind of like this kind of sweet spot here. You need a big sample size to find those areas. So here's just a plot of different GWAS studies that have been done on BMI, height, waist-hip ratio adjusted for BMI. So just anthropometric traits, shape, things having to do with the shape of people's bodies, which are very easy to measure because everyone has a BMI. You give me 10,000 people, you could calculate the BMI for every one of those people, and it's very, very simple. You just take the weight and the height and do some math on it. So these are the ones that tend to have the biggest sample sizes. You can see that, let's take BMI for, I can't tell what color this is because I'm colorblind. Okay, green. Green is height. That's green. So height is very strongly determined by genetics. It's very heritable, as you would see if you've ever looked at a tall family or a short family. And so on the y-axis, we have a number of loci or variants that have been discovered that have been linked with height. And then on the x-axis, we have the sample size on a log scale. So each point represents a single study. So if you're looking at a sample size that's less than 100,000, you'll find dozens, maybe 100 at the most, variants that are linked to height. Once you get over 100,000, you start to get more and more. So by the time you get up to, close to 1 million, you can discover 2,500 or more variants. And of course, I think the number would just get bigger the more you added. And the same is true for both BMI and BMI and waist hip ratio adjusted BMI. So the more people you study, the more variants you can find. And there may be no upper limit to this, really, if you think about it, because you could study millions upon millions of people and probably find more and more variants, especially as you got into looking at other populations where the genetics of height might work a bit differently. Now, there's kind of a flaw to this, or maybe an unfortunate side effect, which is that it's some conditions you just are never going to get that big of a sample. You'll never get a sample size big enough to be able to know for sure if you found all the variants that could be there, because there just aren't enough people with a given condition. So for example, I keep going back to schizophrenia, but schizophrenia, the prevalence is about 1%. So it's gonna be very tough, I guess, to find millions of people with schizophrenia that you can genotype. It's just too hard to do, at least without international cooperation. There's not 1 million people with schizophrenia in Canada. That would be, that would not be possible. So it's not. So sometimes that a condition is probably genetic. You can tell sometimes from twin studies. Twin studies will tell you with a fair degree of certainty whether something is or isn't genetically linked. But you may never have enough people on the planet to tell you what all the genes actually are. So that's kind of a funny irony. This is why we found so many variants and genes that are linked to height, because height can be measured in everyone. You don't have to pick tall people and short people. You just pick everyone of every height that you can find, and then just plot a regression based on the variance in the height. So that's easy. But when you're talking about case control studies or things that are kind of rare, that's very tough. So that's kind of the conundrum we find ourselves in, is we know that the variants are out there. We know that we know kind of how we would discover them, but because there's not enough people on Earth that have that condition, we'll never know for sure what's going on. Okay, so I'll pause briefly. That's all I wanted to say on this topic. These are just some kind of basic facts, and again, I'm trying to maybe expand your consciousness a little bit when it comes to thinking about genomics or genome-wide studies. And, because we'll be looking at a lot of them, and I don't want to have to re-emphasize these points every single time. What I'd like to do is just give you, a framework for looking at these things and understand what the major challenges and pitfalls and, positive and negative sides to them are. Okay. So if everyone's happy with that, we can get on to the review section of the class. So what I'm going to do is just go through and kind of revisit a few slides from the various lectures that we've had so far that I think warrant perhaps another go around on the explanatory front. And of course, if you have any questions, now is the time to start asking them about content, or maybe you just wait till I get to the lecture, whatever it is you prefer. The way I picked these slides, one of the good things about posting these on YouTube is that I have a lot of analytics at my disposal. So I can actually see where people are watching the lectures and what parts they pause and return to and look at multiple times. And my assumption would be that if you look at a section multiple times, it probably means that part was confusing to you, or that you might need more explanation, or maybe I just didn't explain it well, and maybe I can try again. So that's one of the fun things about posting these on YouTube that I can look at. I can look at audience retention too. So of course I can tell exactly when everyone stops listening to me, which is very funny. It's kind of, in the, when you're teaching in real life, there's a certain moment about 5 minutes before class ends when everyone grabs their books and starts packing things into their bag. And there's kind of an equivalent for that in YouTube, because you can actually see your viewership drop off as the lecture goes on. So that's just, that's always, this is why YouTube, professional YouTubers tend to go insane after a while, because they're so fixated on optimizing the analytics that their titles get more and more clickbaity, and they become more and more fixated on having the video flow just right to maintain audience retention, because good audience retention is a key element of the YouTube algorithm that determines whether or not your video will be served up to other people. Anyway, sorry for the rant. I find the YouTube thing quite fascinating. The anthropologists are going to have fun looking at how what it did to our society anyhow. So let's go to lecture 1. So I wanted to go over this slide again. This was talking about Mendelian inheritance as a guiding principle. And what I meant by that is not so much that, we hold it near and dear to our heart and it sort of is our inspiration, but more to say that as a mode of inheritance, the way that inheritance was described by Gregor Mendel is the way that inheritance works across the genome. So for any given variant where you can have multiple alleles, the way that it is inherited is always according to the kind of rules of Mendelian inheritance. The difference is that most variants that we know about now have almost no observable effect on phenotype. They don't fall into this dichotomy of dominant versus recessive, and so you can't look at them using the kind of the classical genetic methods that Mendel would have used and people in it walking in his footsteps would have used. That's what I mean by this. This is the way that inheritance is done. And I guess, I want to point out that this was issued as kind of a contrast to blended inheritance, which I think I described it as sort of as an analogy to color. If you imagine, the blended principle would be that if you take a white flower and a purple flower and you mix them together, you'd get a lighter purple flower as a result with the white and the purple mixing together. But more important than that, you wouldn't be able to then separate out white and purple flowers from them. They would just be forever kind of retain that color that they had that was a blend of the two. And the analogy I thought of, or I think I did mention this, was that inheritance could be a bit like Mendel's description of inheritance being discrete could be a bit like the way Newton described light, which is white light is actually made up of a spectrum. It's made up of the entire rainbow, and so you can use a prism to take white light and separate it into its constitutive colors. You can make white light out of just red, green, and blue. And you can also recombine those back into white light. This is kind of the idea of Mendelian inheritance, is you can have a purple flower, but within it there could be a dominant purple allele and a recessive white allele. And if you bred it carefully, you could re-extract that white allele and recreate a line of pea plants that breed true and always produce white flowers. So that would be analogizing it to light. The contrast would be analogizing it to paint. When you mix paint together, it's permanently mixed together. You can't take red, yellow, and blue paint, mix it together, and then separate them back out into their constituent colors. It's not physically possible. So that's all I really wanted to say there. And so, the simple rules of Mendelian inheritance don't apply to most traits, because traits are typically polygenic, meaning that they're made up, or your status on a given trait is determined by a whole combination of variants that all kind of add up to whether or not, to where you fall along that spectrum or on that trait. So traits are typically polygenic, and the rules of Mendelian inheritance don't exactly apply. However, for each of the individual alleles that make up the trait, the rules of Mendelian inheritance do apply. They're inherited in the same way that Mendel described. If you wanted to, you could breed people or mice or whoever that had. You could take a heterozygous mouse and you could find a way to breed out one of the two alleles and have mice that were breeding true for one of the two alleles, even if they were mixed together in your founding population. That's all I'm saying. So there's kind of a difference, I suppose, between traits and alleles when it comes to whether or not the rules of Mendelian inheritance apply. And I also want to talk a little bit more about the Hardy-Weinberg equilibrium. This is a concept that you may have remembered from your genetics classes, and it's really a pretty simple concept, but I think it's quite a powerful idea. The Hardy-Weinberg principle is simply this. Allele frequencies in a population will remain stable unless some kind of selection is operating. So selection would mean differential survival or differential reproductive success. Selection is the basis of evolution, right? Evolution is the non-random survival of randomly varying replicants. So if survival is completely random, then evolution can't happen. But if evolution has a selective power, meaning that people organisms with certain variants do a bit better than organisms or other organisms, then the genes that are carried by the more successful organisms will tend to become more frequent in the gene pool, and the ones carried by the less successful ones will become less and less frequent until they may disappear altogether. So the Hardy-Weinberg principle just says that as long as selection isn't happening, allele frequencies are going to remain constant across generations if the population you're looking at is big enough. So what this is really used for in our world is to ask or to see if we can determine empirically whether selection is happening at a given locus or for a given allele. And I haven't given you the math of it here, but essentially what the equation, the Hardy-Weinberg principle is based on, allows you to take the allele frequency of a given genotype, like say you know the number of people you have or the number of animals you have that are homozygous for the minor allele, the recessive allele, in this case, AA. If the population is in Hardy-Weinberg equilibrium, then from only knowing that, you would be able to determine the frequency of the heterozygous genotype as well as the homozygous dominant allele. You would be able to predict how many people or how many specimens you should find with those two genotypes as well. And that gives you a hypothesis to test against, because you have a prediction of how many should be out there with that genotype. But if you find that there are more or less of people with a certain other genotype, what it would imply is that that particular allele is not in Hardy-Weinberg equilibrium. What this means is that some form of selection is operating on it. And this is just a pretty useful piece of information, because if you have like for example, if you have a variant that might be linked to a disease, what you would expect, given that it's causing a disease or linked to a disease, you would expect it to be selected against, right? Because why would natural selection favor a variant that causes sort of a condition? People with diseases don't tend to do as well than people without diseases. So you would expect disease-causing alleles to be selected against. If a disease-causing allele is in Hardy-Weinberg equilibrium, it means that somehow it's not being selected against by evolution, which is a pretty interesting thing to consider. Why would, for example, a gene that causes, say, autism spectrum disorder not be, why would a gene such as that be in Hardy-Weinberg equilibrium? Could it be that while the gene can cause problems if you have it combined with a bunch of others, on an individual level maybe it's actually helpful or at least neutral? That's the kind of thing you can ask when you have this type of information. So it's really just a fact about the way that allele frequency is spread across a population that can let you estimate whether a given allele is being selected against or not. And then from there, you can think about all kinds of fun, interesting interpretations. So now let's go to lecture 2. This is a slide that people came back to a few times, so I wanted to go over this another time. Here I was giving you a... so I just got a question here. Let me jump back. Nathalie asks, I'm having trouble reading the graph, specifically the y-axis. So if the allele frequency for the P allele is 0.6, the allele frequency for Q is 0.4 in the population. Let's go back to that slide. Yeah, so that's exactly correct. So this is what the allele frequencies would look like if a given variant is, or if a given variant is in Hardy-Weinberg equilibrium. So if you knew, for example, that the frequency for the P allele was 0.6, sort of mathematically you would know for sure that the Q was 0.4 because they have to add up to 1, right? You can't have something that's more than 100% frequent. What's more interesting is that if you look at the position of these different lines. So if the particular alleles are in Hardy-Weinberg equilibrium, and you know that P is 0.6, the frequency of the AA genotype, the homozygous for the Q allele, would be represented by the blue line. So that would be equal to about, I can't tell what that's, 1.9, let's say. The frequency for the homozygous dominant allele, AA, would be a little less than 0.4, and the frequency of the heterozygous allele would be about 0.5. So this gives you like a prediction, basically. Like if you know that, if you know any of these pieces of information, you can essentially solve for the other ones. If you know, for example, that AA is equal to is frequent is a 0.0 no 0.19 percent of the population, then you would be able to assume that the other alleles have these frequencies in the population. So this is just like a math equation you can solve for. And so that's the assumption that you would get if everything was in Hardy-Weinberg equilibrium. If it wasn't, these lines would be in different places and you would be out of equilibrium, and that would give you a sense that there's some kind of selection operating. Ali asks, isn't the problem with natural selection that it only selects for traits that increase the chance of survival? If a disease trait doesn't affect survival, it wouldn't be selected for. That would be the idea, yeah. So the only thing natural selection can really operate on are your odds of reproducing. Really, that's what actually ends up mattering. Do you or do you not pass your genes on to the next generation? If your genes make it easier for you to pass them on, like if your genes are easier to pass on to the next generation, either because you survive longer or because you have better reproductive success some other way, then they tend to increase in the population theoretically. So if a gene is completely neutral with respect to either survival or reproduction, I mean reproduction is central. If you survive, actually kind of doesn't matter. You can, as long as you reproduce, you're good. If you die the second after you've reproduced and your offspring survive, that's just fine, I guess. Which makes you wonder what the point of grandparents is, but we all love our grandparents, don't we? Anyway, so if a given trait has absolutely no effect on survival or reproduction, then it would be basically neutral with respect to natural selection. And that's actually what's going on with most of the genome. Almost all of the genetic variation that exists between human populations has nothing to do with natural selection per se. It's really more genetic drift. Humans will populations will tend to drift genetically as certain alleles fall in and out of favor for purely random reasons. This, by the way, is kind of contrary to the Hardy-Weinberg principle, but most of the variation in populations comes from that, which is kind of an important point actually to bring up when you're dealing with like those kind of like racist people who are like, different populations are kind of intrinsically better than others at certain things. Most of the variation between populations has nothing to do with survival or like what environment they evolved in. It just is because of genetic drift. If you take two groups of people and put them on separate islands and leave them separated for 10,000 years, they're going to develop all kinds of genetic differences because they're an isolated population that are breeding separately from another isolated population. And those differences probably have nothing to do with how well it helps them survive. It's just random drift carrying them in one direction or the other. They would come back after 10,000 years of being separated and look and sound and appear to be somewhat different, but almost none of those variants would have anything to do with survival. That's just the way evolution works in humans. So you can use that as an answer to people like Charles Murray or those kinds of characters who are arguing that like, certain people are better at certain things because of their ancient traditions or whatever. Yeah, it's not how it works. Back to this slide. So the variation in biochemical pathways, I was really just trying to point out that like this is one of the types of studies that was done quite often when you had variants that were located in biochemical pathways that you already knew about. For example, monoamine or catecholamine synthesis or breakdown. You could study those and link them to a trait with the kind of knowledge that every piece of information you got could be tied to a given thing happening in the brain. So the COMT Val-Met allele is just one of these that was very widely studied. It's a variant in the gene COMT, catechol O-methyltransferase, that converts the 158th amino acid, which is normally valine, to methionine via a coding SNP that changes an A to a G. This is changes the changes the codon and makes the codon code for a different amino acid, and it basically just makes the enzyme better or worse at breaking down dopamine, and thus the people would have theoretically either higher or lower rates of dopamine turnover, and because dopamine does so many important things in the brain, it would in principle change their behavior and change the way they think about things. So that's all I was kind of getting at here. The frequency for the different alleles is given in a variety of populations here. You can kind of see that, like, either due to selection or genetic drift, People from like Europe, for example, CEU, have different frequencies of the different genotypes than say people from China, the Han Chinese, CHB, and everyone else here. And it's difficult to say if this contributes to any meaningful personality differences across the different racial groups. I would imagine most likely not, because these variants exist in the context of many other things, and it's hard to say if this would even be if this would even matter in the grand scheme of things. But like many alleles, there are differences in the frequency across population groups. And then this one, I also noticed people were looking at a fair bit, and this one I think is hopefully. I'm not trying to, this might have been marginally confusing, but the basic idea is this, and maybe this will make more sense now that we've talked about DNA methylation in a more proper way. DNA methylation is measured by the percent of methylation on a given cytosine in a CG dinucleotide or a CpG dinucleotide. So it can go between 0 and 100%, with 100 being fully methylated and 0 being fully unmethylated. Most places in the genome are, fairly highly methylated, between 70 and 90%, except for CpG islands that are closer to 0% down there. And methylation varies for a number of reasons. It varies because you're in a different tissue. It's epigenetic, of course, and different genes are expressed in different tissues, so methylation would vary across tissues. It also varies across time points. Sometimes when you're developing, you have fairly low levels of methylation, and as you become more mature, and become more developed, the methylation levels tend to get higher as your epigenome reaches a mature state. And so there are those factors that influence DNA methylation, but as it turns out, genetic variation can also influence methylation. And so what I was pointing out here is what's called an MQTL, a methylation quantitative trait locus, where the genotype that you have at that particular SNP, this SNP is RS2108854, determines or contributes to determining the amount of methylation you have at a nearby cytosine. So here, there's a kind of a relationship between the number of copies of the A or G allele you have and the methylation you have in the fetal brain. So the more copies of G you have, the more methylated the DNA is in the fetal brain. If you have 2 Gs, you're close to 60% methylation. If you have 2 As, you're below 50%. So there's a fairly sizable difference there, depending on the number of copies of the allele you have. But what's important to mention is that because methylation is tissue and time point specific, your allele status makes no difference in the prefrontal cortex of an adult, nor does it make a difference in the striatum, but it makes has the opposite effect in the cerebellum. So what I'm really trying to say here is that DNA methylation is a very important epigenetic feature that is related to a lot of different things, and it's determined by many factors, including genetics. So there's this interaction between genetics and epigenetics that means that, if you have genes that put you at risk, for example, for something, those can be added on top of the epigenetics that may also put you at risk. So you can have kind of, in principle, I guess you could have a risky genotype as well as a risky epigenotype, and the two of them synergize to make things even worse for you. Or you could have a scenario where your genotype puts you at risk, but your epigenotype compensates for that by being the opposite. And because the epigenetics are modifiable by the environment, this might give an explanation for why people with the same genes have different disease risks depending on what kind of environment they grow up in. So very kind of broad point to be made from just one single box plot, but I think it's worth pointing out that genetics and epigenetics are interlinked. They one affects the other and vice versa. So here's lecture 3. This is the microarray platform that I was talking about. And this is how genotyping is done for GWAS studies. The basic principle is this. You can fragment people's DNA into a bunch of small little pieces, about 100 base pairs in length, fluorescently label them so that they glow in the dark, and then expose them to a microarray, which is covered in thousands or millions of probes. And the probes are designed such that they're complementary to areas of the genome that flank variants. So you have a variant, a SNP that's located in some sort of place, and then you have the sequence that flanks it upstream and downstream that kind of gives it a unique identity. If you just had one SNP, you just have an A or a G or a T, you'd never know where it belonged. But you have a flanking sequence that helps it be located. And then when you expose the fluorescently labeled DNA to the microarray, where it binds tells you what SNPs the people have. So if you have somebody who's homozygous for, say, the minor allele, their DNA will only bind on the probes for the minor allele, and you'll see no fluorescence coming from the major allele. If they're heterozygous, you'll see a bit of fluorescence coming from both probes, and if they're homozygous for the major allele, you'll see all the fluorescence coming from the major allele probe and none coming from the minor allele. So you take this concept of like the DNA can bind to specific places based on whether or not it matches. You know, if there's a SNP that's missing, it won't bind, and if the SNP is there, it will bind. You take that concept and you repeat it millions of times, you can determine the genotype of millions of different SNPs in the person in the person you're looking at. It's kind of like you know those coin sorter machines that like you pour a bunch of change into the machine and it'll sort out all the quarters and the nickels and the dimes and whatnot. It's kind of doing that, but for SNPs. So lecture 4. This slide was just to point out that gene expression is ultimately kind of the, you know, for our purposes at least, is the final common pathway at the end of all of this epigenetic regulation, or genetic and epigenetic regulation. Ultimately, what matters is, you know, assuming the sequence of the gene is the same. As we said, most variants don't affect the protein coding sequence. What really matters then is how much of the gene is expressed and when it's expressed and in what quantities and so forth. So the amount of expression is very important, and I wanted to just emphasize that expression is typically regulated by the binding of proteins such as transcription factors to binding sites on the DNA. So the transcription factors actually possess binding domains on them that are complement or that are able to locate and identify specific sequences in the genome. And wherever they happen to find those sequences, they just stick on. And once they've stuck on, those proteins can recruit other things like RNA polymerase and bring a whole bunch of the transcriptional machinery to bear on the gene. So it's this interaction between the binding domain of a transcription factor and a complementary or like a specific sequence in the DNA that enables this machinery to work. If a transcription factor can't bind, it's not, you know, the gene won't be expressed typically. And of course, you can also have repressors. These are just basically transcription factors that do the opposite of activating it. You know, typically, I think I mostly consider them to be most transcription factors I would think of as activators, but there are also repressors where if they bind, they stop transcription. These could be like the polycomb proteins, for example, bind to genes and prevent them from being transcribed. And those would essentially just block RNA polymerase from binding, thus making it impossible for RNA to synthesize. So it's this protein binding with the genome that regulates expression and whether or not genes are turned on or off or not. So this is an element where there's a lot of room for epigenetics to get involved and change the ability for transcription factors to bind or not bind. It also depends on the three-dimensional structure, right? If you take these transcription factor binding sites and put them into heterochromatin, where they're all packed away and bundled up, the enzymes and the transcription factors can't get in there to bind, so the gene won't be expressed. And here I just wanted to point out that like the picture I gave is a little simple. In reality, transcription factors work in a combinatorial fashion, sometimes involving regions of the DNA that are rather far away from the transcription start site. These would be like enhancers. So the overall picture is a good deal more complicated than that, but the simple principle is this. Binding of proteins like transcription factors to the DNA is what ultimately regulates gene expression. Gene expression is what ultimately determines phenotype. And the ability for these proteins to bind is something of an epigenetic phenomenon because it depends on, you know, the shape of the chromatin and how much DNA methylation there is and histones and so forth. It's also genetic because if you have variations at these transcription factor binding sites, then these transcription factors can't bind the same way, and that can change the way genes are expressed. Okay, so lecture 5. Here's the epigenetics lecture. So I wanted to kind of go over the idea that CpGs are rare, and this is a fairly common way of thinking. When you're talking about whether something is rare or common, you want to have kind of a baseline to compare against, and the baseline is typically how would things look if everything was completely random. If you take the 16 possible dinucleotide pairings, AT, AA, AC, AG, all the different ways you could put all the 4 letters of DNA together into pairs, there's 16 of those in total. And you would expect that given that Gs and Cs make up about 42% of the nucleotides in the human genome, that the combination of C followed by G would happen about 4.41% of the time. This is just kind of a mathematical reality of how many Gs and Cs there are. You'd expect you'd expect CG to be 4.41%. You'd also expect GC to be the same percent. So this would be the expectation. However, when you actually look at how often CpGs appear, they're way, way less common. They're present about 1% of the time when you'd expect them to be present over 4% of the time. So that's all I was kind of getting at there, is that whenever you see something in the genome where something is like missing or not present in the frequency that you would expect, that tells you that there's something up. It tells you that there's something happening there, either over the course of evolution or what have you, that's causing this deviation from the expectation. And CpG islands are an exception to this, where you actually have a ton of CpGs. Now, methylation there are CpGs throughout the genome, not at CpG islands, but CpG islands are just where a lot of them are concentrated. And CpGs that are located throughout the genome are typically highly methylated, but those that are located in CpG islands are typically have very low levels of methylation. So that's a funny kind of dichotomy there. The places that have the most CpGs also have the least amount of CpG methylation. Okay, and so I wanted to also talk about histone modifications one more time. These are a lot more complicated in a way than DNA methylation, because there's really only one kind of DNA methylation. It happens at CpG dinucleotides, it happens to the cytosine in those dinucleotides, and it can be described as a percentage, either 100% meaning all of the cytosines in your sample are methylated, or 0 meaning none of them are, or somewhere in between. When it comes to histone modifications, you are talking about a much more complicated scenario, because there are 4 types of histones that are involved in the nucleosome. The nucleosome is what the histones form into, around which the DNA is wrapped. All of these histones have tails on the N-terminal end, and these tails have many, many amino acids, each of which could potentially be modified in some way that changes the way the histone works. So this is not the kind of thing I would want you to remember, but you can have, for example, H2B is one of the types of histones. You can have acetylation either at K5, K12, K15, or K20, or all of them, or none of them, any of the 4 of them combined. Any of those are possible, and that's just one type of histone. You can have methylation at the H3K4 locus. That's the 4th lysine. You can also have methylation at the 9th lysine combined with acetylation. Sometimes histone methylation causes an increase in expression. That's permissive methylation. Other times it causes a decrease. This is called repressive methylation. So this is just, all I'm saying here is that there's a lot of different kinds of histone modifications. They're very complicated. There's a certain logic to the name that tells you what histone is being modified and how it's being modified. But beyond that, there's no rule that I can give you that tells you what always, acetylation always does this, methylation always does this. Not the case. And this is something I added to, I want to just be really crystal clear about this, because I think this is a somewhat confusing thing that maybe I'm not helping overall by the way I talk. But there's a very important difference between DNA methylation and histone methylation. So just read that through again. DNA methylation versus histone methylation. 2 separate things. The fact that they both have the word methylation in them is only because methylation is one of the most common types of chemical modifications that can happen in the body. COMT, remember, catechol-O-methyltransferase, that's actually a dopamine methylase. What that does is it adds methyl groups onto dopamine, the molecule, the neurotransmitter dopamine. It adds methyl groups onto dopamine, and that's one of the first steps in breaking down dopamine. So methylation is a very common biochemical reaction. What it does to gene expression varies entirely depending on what's being methylated. So DNA methylation is a chemical modification of the DNA molecule itself. It's happening when a methyl group is added to the 5th atom of the pyrimidine ring of cytosine. So it only happens to cytosines, and typically in CpG dinucleotides, although it can also happen at CH loci, mostly in the brain. So this is carried out by DNA methyltransferases, and almost always DNA methylation represses transcription. Not every time, but enough that it's basically a good rule to follow. DNA methylation represses transcription. histones are not DNA. Histones are proteins that organize the DNA. So the DNA is like wound around histones in the same way that like your headphone cables might be wound around those like plastic gizmos that people have, or like your speaker wires might be wound up around zip ties or something. So histones are proteins around which the DNA is organized. And so histone methylation is the chemical addition of a methyl group to the tail of a histone. And histone methylation does not always decrease expression. It can have varied effects. Sometimes it depends on how many methyl groups are added. Sometimes it depends on what histone they're attached to or which amino acid residue in the tail they're attached to. So very big difference there. And I think that hopefully should clarify things a little bit. You can just look at this table to see why I won't give you a hard and fast rule about histone methylation, because look, histone H3K4 monomethylation activation, H3K9 monomethylation activation, H3K4 dimethylation repression. But then if you go up to trimethylation, 3 methyl groups, you get activation. So 1 group on the H3K4 activation, 2 methyls repression, 3 methyls activation. So it's very complicated. There's no real hard and fast rule that I can say that will give you that'll always tell you what to look for. If you ever get curious, you can come back to this table. It's in the epigenetics lecture, but generally this isn't the kind of thing that I would want you to memorize because I don't even have it memorized. Okay, so I can see we're out of time here, so we'll end there, folks. I hope that was helpful to you. Like I say, I haven't. It's a little tough for me to tell what you are or are not finding easy or understandable here. My goal is to try to help you understand as much as I can before the quiz on Monday. The way I've been doing it, in the absence of any questions or feedback, is just to look at the YouTube videos and see where people keep looping back to and trying to, perhaps having trouble with. So that's what I'll be doing, going forward. If you have any questions, please feel free to ask or come to the office hours tomorrow. I think that'd be a good opportunity. And otherwise, just have fun studying, folks. It's feel free to rewatch any of the lectures if you like, and see if that helps. And again, I'm always here if you have questions. So with that, I will say goodbye, and I hope that you have a good rest of your day. And we'll see you tomorrow at office hours. Okay, so long.